Let's jump right into this explainer, okay? Today, we're uncovering these invisible, literally life or death water movements that are happening inside every single living cell in your body right now. I mean, we're talking about a biological process so fundamental, yet so incredibly hidden, you probably don't even realize it's keeping you alive at this exact second. So, here's our quick roadmap for today. We're going to cover water as the ultimate solvent, the basics of osmosis, and how to investigate it in action. Then we'll get into understanding water potential, looking at plant cells under pressure, and finally, osmosis in animal cells. Let's get to it. Kicking things off with Section 1, water as the ultimate solvent. You see, to really understand osmosis, we have to establish right up front that water isn't just, something you drink to quench your thirst. It is the ultimate biological transit system. There's a very specific reason it's the most abundant molecule in the human body. It's actually amazing how many roles water plays just by being an excellent solvent. Think about it. In us humans, it creates blood plasma, which is absolutely crucial for transporting carbon dioxide, nutrients, and hormones all over the body. In plants, it moves dissolved minerals up through the xylem and glucose through the phloem. And for animals, water literally breaks down food through hydrolysis, so molecules can actually pass through the intestinal wall. It even dilutes toxic waste products like urea, safely flushing them right out of our systems. It's a total game changer. Moving on to Section 2, the basics of osmosis. So now that we know water transports practically everything, you might be wondering, how exactly does it get in and out of our tightly sealed cells to actually do all this work? Well, that brings us to the real hero of today's explainer, osmosis. By definition, this is the diffusion of water across a partially permeable membrane, moving down a concentration gradient. Actually, I've always found it super helpful to think of that cell membrane like a really exclusive biological bouncer. It allows certain molecules, like water, to just breeze right through the VIP line, but it completely blocks the bigger ones. The absolutely crucial thing to grasp here is the gradient itself. See, a dilute solution actually has an abundance of free-moving water molecules, but a concentrated solution, think like really salty water, has way fewer free water molecules. Water is always going to flow down this gradient. It moves from that crowded, free-flowing dilute side over to the concentrated side, just desperately trying to balance the whole shebang out. Now, this all sounds great in theory, right? But you're probably wondering how scientists actually prove that these invisible, microscopic molecules are shifting around. I mean, how can we actually see this in action? Well, that takes us straight into the laboratory. Which brings us to Section 3, investigating osmosis in action. Let's pull this concept right out of the textbook and look at two classic experiments that bring it vividly to life, right in front of our eyes. Okay, so in our first setup, we use this stuff called partially permeable dialysis tubing. It perfectly mimics a living cell membrane. We lock a really strong sugar solution inside it, secure it tight to a glass capillary tube, and then dunk the whole thing into plain water. And get this, within just 10 or 15 minutes, you can literally watch the liquid defy gravity. It starts rising up the glass tube as the water from the beaker forces its way inside. It's so cool to see. For our second experiment, we're using actual living tissue. We take a cork bore, cut 5 identical potato cylinders, weigh them, and then dunk them into 5 completely different sucrose solutions. We let them soak for a good 30 minutes. After that, we dry them off and weigh them again to track their exact percentage change in mass. And the results brilliantly illustrate osmosis in action. The potatoes sitting in the weaker solutions actually gained mass. They felt incredibly firm because water just rushed into their cells. On the flip side, the ones bathed in the concentrated solutions lost mass and went completely limp because all their internal water got drawn right out. All right, moving into Section 4, understanding water potential. For those of you diving a bit deeper into the science, we really need to elevate our vocabulary a notch. We need to understand the underlying mechanics of exactly why this water movement happens the way it does. You see, instead of just using the word concentration, scientists use a much more precise term, and that's water potential. It's officially defined as the net movement of water molecules from a region of higher water potential to a region of lower water potential, straight through that partially permeable membrane. Let's think about the physical reality of this for a second. A high water potential basically means there's a really high proportion of free-moving water. But a low water potential? That means it contains dissolved substances, like sugars or salts. And these dissolved molecules act almost like microscopic magnets, grabbing onto the water molecules and physically stopping them from moving freely. Keep in mind, though, molecular movement is absolutely a two-way street. Water is constantly buzzing in both directions across that membrane. But because the dilute side has so much more free water, the overwhelming net traffic just surges toward the lower potential. The larger sugar molecules on the other side, they just diffuse way too slowly to counteract that massive rush of water. Section 5. Plant cells under pressure. So how does this microscopic traffic jam actually affect a living organism? Well, let's take a look at how plant cells uniquely utilize this pressure just to survive out in the wild. Plant cells are super interesting because they're trapped inside these really rigid, inelastic cell walls. When water rushes into a plant cell via osmosis, the vacuole inside expands and pushes incredibly hard against those walls. This creates what we call Turgor pressure. This pressure makes the entire cell rigid, or turgid, which is exactly the structural support that keeps a plant's stems and leaves proudly standing upright, reaching for the sun. But, the reverse is also true, and it's highly dangerous. If a plant cell is bathed in a solution with a lower water potential, water rapidly escapes from the vacuole. The cell loses all its turgor pressure and basically just goes flaccid. This process is called plasmolysis, and on a macro level, it's the exact reason your thirsty houseplant wilts and droops when you forget to water it. The physical stakes for a plant cell locked in that rigid box are just massive. Just look at the difference. On one hand, a healthy turgid cell has a completely full vacuole, pushing outward with immense strength. On the other hand, a flaccid cell has this contracted, sad-looking little vacuole, offering absolutely zero structural support. Okay, final section. Section 6. Osmosis in animal cells. Seeing what plants go through naturally leads to a pretty alarming question, doesn't it? What happens to organisms that don't have those rigid, protective cell walls to handle all that intense internal pressure? Well, animal cells, like, the ones inside you and me, are incredibly vulnerable. Without a cell wall, the fluid our cells are constantly bathed in absolutely must have the exact same water potential as the cell contents. If our blood plasma somehow gets a higher water potential, water is going to rush in, and without a wall to stop it, pop. The cell literally violently expands and bursts. No way around it. And if the fluid has a lower water potential, the cells become plasmolyzed, shrinking all the way down, and their critical functions get severely impaired. To prevent this microscopic disaster from happening every time we drink a glass of water, our biology is working constantly in the background. Your brain is always monitoring, and your kidneys are always adjusting the water potential of your blood. It's this incredible, continuous, subconscious dance just to keep our tissue fluid perfectly balanced, making sure our cells neither swell up and explode nor shrink down into nothing. So as we wrap up this explainer, I want to leave you with one final thought to chew on. If your kidneys are constantly, silently adjusting the water potential of your blood just to save your cells from bursting at this very second, what other invisible, microscopic balances are keeping you alive right now? It's honestly mind-blowing to think about. Thanks so much for exploring this fascinating topic with me today, and hey, keep questioning the hidden world inside you.